Hello, good morning, good afternoon, good evening. Welcome you all. Today we join here for our second Going Organic seminar series for the spring 2022. Thank you all for joining around the world. Our partners from India, Rwanda, Africa, parts of Africa, and Canada and US. Thank you for all of you joining. My name is Dil Tavaraja. I am Associate Professor leading the Clemson University Pulse Quality and Organic Nutritional Breeding Program. And our program is, and especially the Going Organic online platform, is sponsored by several funders. The first agency is the United States National Department of Agriculture, NIFA ORAI, and USAID Feed the Future Innovation Lab, located at the Cornell University, South Carolina Department of Agriculture, the Good Food Institute, and the Global Food Shot, and the USDA Agriculture Research Services. I thank you all the funders for the program, and we reach out to the growers, researchers, scholars, and young breeders who wants to be in high school, even undergraduate and the graduate students. And thank you for all support. And now I'd like to introduce Mr. Mark Dempsey. He's a PhD student at my program at the Pulse Breeding Program. And he will be introducing our guest speaker, Dr. Audrey Kalil. Thank you, Mark.
Yeah, thank you, Dr. Dill. Welcome, everybody. It is my pleasure to introduce today Dr. Audrey Kalil, who earned her BS in biology from the University of Minnesota Twin Cities and a Ph.D. in plant pathology from the University of Wisconsin-Madison. Her grad research focused on host genetic signaling pathway required for the establishment of rhizobial and arbuscular mycorrhizal symbiosis in 2 model legume species. And after that, in 2015, she joined North Dakota State University as a plant pathologist at the Williston Research and Extension Center, where she leads an applied research program focused on integrated pest management of diseases affecting cool season pulses, durum wheat, and sugar beet, as well as the enhancement of rhizobial symbiosis in pulse crops. And so, Dr. Kalil, we're very happy to have you here today, and I'm going to go ahead and hand it off to you.
All right. Well, thank you for the introduction. Thank you for having me this morning. Let me share my slides real quickly. Okay, can everyone see?
Yes.
All right, great. Okay, well, good to be here. Thank you all for attending this morning. I'm based out of the Williston Research Extension Center, so far northwest North Dakota. If you're familiar with North Dakota State University, the main campus is in southeast North Dakota. And so we're about 6 hours away from main campus in the heart of pulse growing country. And I'm going to discuss some of the primary diseases that we have in our growing region. So I think this audience is familiar with pulse crops, but just in case you're not, it's important that we define and distinguish the pulses from other legume crops. And so these are crops that are harvested primarily for the dried seed, rather than like fresh beans or fresh peas. And they're primarily consumed, it's for human consumption rather than crushing, as in the case of soybean for the oil. And the pulses can include dry beans, dry peas, chickpeas, and lentils. And I'm going to be primarily discussing the dry peas, chickpeas, and lentils. So just to give you an overview of where pulses are grown in the United States, you can see that I highlighted with the star the Williston Research Extension Center. So you can see there's a lot of pulse production in North Dakota and Montana, a lot of acres in the Palouse area of Washington and Idaho. But there is pulse production scattered around the US, and it's growing because of increasing demands for some of these pulse products, high protein sources such as processed peas. And, the areas where we've really seen these pulse acres take off are semi-arid, and the pulses are finding a niche in the rotation in between wheat. And so there were a lot of wheat fallow acres historically, kind of preserve limited moisture, but the pulses fit nicely into this rotation. They can take advantage of what little rainfall occurs, and they're well adapted to no-till, which has become predominant along with the incorporation of these pulses. So we've seen a big shift from wheat fallow with a lot of tillage to pulse wheat in no-till conditions. So it's been a really big benefit environmentally in this growing region. Unfortunately, of course, no benefit comes without its downside. So the diseases I'm going to focus on are those that affect us here in North Dakota. And I broke them into 3 different categories. So we have our seed rot and seedling blights. So these are diseases which can directly rot the seed in the ground or cause the plant to die shortly after emergence. Multiple soil-borne fungi can contribute to this disease, but I'm going to focus on Pythium, which is an oomycete soil-borne pathogen, as it can be very problematic in chickpea. Then I'll discuss 2 root rots, Aphanomyces root rot, which affects pea and lentil, and Fusarium root rot, which can affect all 3 of these pulse crops. So Aphanomyces eutyches is an oomycete pathogen, and Fusarium root rot is caused by multiple species of Fusarium. And then I'll finish off with foliar disease with a focus on Ascochyta blight. Again, this is a disease which is very problematic on chickpea. It's probably one of the most yield limiting diseases in this crop. It can affect all 3 of the pulse crops, but in a host specific manner. So I've listed the pathogens there. So Ascochyta rabii affects chickpea, Ascochyta lentis affects lentils, and then there are 3 different pathogens which affect pea. One of them, Mycosphaerella pinodes, is a reason why it's sometimes called Mycosphaerella blight as well in the case of pea. So I'll start off with Pythium, seed rot, and damping off. So you can see in this image with the healthy pea on the left and the rotten peas on the right, it's kind of, the seed looks a little bit brown, and that's because the soil is sticking to the rotting seed. So the seed itself can become kind of gelatinous and completely collapse in the soil. Sometimes the plants can emerge and then die shortly after that. So what you end up with is random pockets of poor emergence at the field level. So this is a chickpea field in the Palouse growing region. That's that area of Washington and Idaho. And you can see the circular dead areas in the field. So where the seed didn't even come up or the seedlings died shortly after emergence. This can be confused with other issues, planter issues or cutworms, for example. Those will typically be in more of a linear formation in the field rather than these circular areas. But if you dig up the rotten seed, that can help you diagnose the causal problem here. So some of the risk factors for Pythium, first and foremost, is high soil moisture and cool soils. So when you're planting pulse crops into cool, wet soils, that's going to be a high risk scenario. Also, anytime you've got compaction, so field edges where you're driving over it a lot, or if you're practicing tillage, compaction can exacerbate this condition because, again, you're having drainage issues. You're having the seed sitting in a wet, cold soil. And then poor seed vigor. So again, anything that's going to delay that seed coming up out of the soil is going to increase the amount of time that it's susceptible to this pathogen. So I'll discuss conventional management on the next slide, just because that's typically how it's managed, a chemical approach in conventional systems. But if you're needing to manage this disease without essentially seed treatments, you're going to want to delay planting until soils are warm. So as chickpea is the most susceptible to this disease, and we're not seeing as much problems with the other root rot, my recommendation in our growing region is to delay chickpea seeding until soil is warm if you're not using a seed treatment. Field selection, so you're going to want to choose a field where there's good drainage. The seed's not going to be sitting in a wet soil, and a field that doesn't have history of disease. So I've had some cases where I plant chickpea with no seed treatment and there's no problems, and other times where I plant chickpea without a seed treatment and nothing emerges. and it's just different fields, presumably with a different soil microbiome, different disease pressure in those fields. So choose a field where you don't struggle with that. And then use vigorous disease-free seeds. So I think Uda from Montana State is on the call, but Montana State University has a regional pulse crops diagnostic lab. You can send seed to them and they can do a screening for different seed-borne pathogens for you. Of course, one with a good germination rate as well, but you really need to give the crop every chance that you can, and so that it gets up out of the ground right away. So I just wanted to briefly touch on chemical management in case anyone, has both growing systems on their farm. And so active ingredients, metalaxyl, mefenoxam are commonly used. They're in different seed treatment packages. Unfortunately, metalaxyl resistance has come up in the Palouse. And so they're using ethoboxam seed treatments with success. Ethoboxam is also registered for control of Aphanomyces. So just be aware that if you're observing, fungicide failures, to get that checked out, work with your local extension to determine if you have an issue with metalaxyl resistance. So far, fingers crossed, we haven't observed that in North Dakota, and hopefully it stays that way. All right, so moving on to Aphanomyces root rot. This is one of the most damaging diseases of peas and lentils. It's become more and more predominant due to short rotations with pea and wheat or lentil and wheat. And these symptoms here are characteristic. So you have this golden brown discoloration in the roots. It travels up into the stem. And if you're handling the roots, they're slippery because the outer portion of the root will just slip off in your hands. If this is if the disease progresses in the fields, the root can turn black, and the stem will shrink a little bit. And so if you're not diagnosing this disease right away, it's very difficult to determine if it's Aphanomyces just based on field symptoms. This is an example of a field in North Dakota with severe Aphanomyces root rot. In this case, plants actually died early in the season, and so you ended up with these large bare patches, and they tend to follow where water is sitting in the field. Again, we have an oomycete pathogen here. There were issues along the field edges where the field was compacted, and water is going to, rainfall is going to spread this pathogen through the field. So this grower did experience severe losses. To diagnose this pathogen, I actually took the diseased plants, planted some sterile pea seeds on top of it, and the root on your right is what the pea plants looked like after growing on those infested roots. So again, it can be difficult to diagnose this pathogen from just looking at field symptoms. So these oospores, the production of oospores is what makes this pathogen really difficult to manage once it becomes established in the field. They can survive up to 10 years or longer. And so once you get a high level of the pathogen in the soil in your field, you need to wait a long time before you can grow a susceptible crop again. And if you're diagnosing this pathogen, identifying these oospores in the roots is going to be a way you can distinguish it from other common root rots like Fusarium. So there is a little bit of host specificity. Lentil and pea are very susceptible. Clovers and vetches can also host. Chickpea is considered resistant, so there is a rotation option in a semi-arid climate. Chickpeas are a really great option to break up the crop rotation with peas and lentils. Resistant varieties of faba bean and alfalfa are available, and soybean, fenugreek, and lupins are considered non-hosts. Even within these susceptible crops, there is a degree of host specificity. So isolates from pea have been found to be weakly pathogenic on bean and alfalfa, and same for bean. Isolates have been found to be weakly pathogenic on pea and alfalfa. So there is some specificity with the pathogens. 2 races can affect alfalfa. Race 1 may be pathogenic on pea, but race 2 does not affect pea. So if you're rotating with alfalfa, it'll be important to select a resistant variety. And if you want to get your soil tested to determine which race is present, the University of Wisconsin has a soil testing service available where you can send soil and they'll determine which race of Aphanomyces is present. So no resistant varieties are available. There has been some work to develop some germplasm with resistance, but currently none of the varieties that are available have any resistance. So in terms of peas and lentils, crop rotation is key. A minimum 4-year crop rotation, so 3 years in between peas or lentils is what we're recommending. If Aphanomyces is not present, if Aphanomyces is present, a longer rotation is recommended, again, because of those long-lived oospores. Field selection, similar to Pythium, you're going to want to choose a field with good drainage, little compaction. For Aphanomyces, this pathogen is actually more vigorous under warm soil conditions. So contrary to the recommendations for Pythium, with Aphanomyces, we're going to recommend early planting. Let the plant have a chance to get going before this pathogen becomes more active under the warm soil conditions. And then there's been some research that a brassica green manure, so growing like a turnip or mustard crop, and then incorporating that into the soil can have a soil fumigation effect for the management of Aphanomyces. And then, or an oat pre-crop, so growing oats prior to a pulse crop has also been found to have some benefit. So if you're practicing tillage, growing a brassica green manure, a brassica cover crop, and incorporating that prior to the peas might be beneficial. So Saskatchewan Pulse Growers has developed this field selection guide that is, I think, really helpful to help determine if a field is a good choice, especially if you're already growing peas or lentils on your farm. And so they have these major influencing factors and intermediate influencing factors for deciding if you're in a higher risk or lower risk category. A positive or negative test result, that's first on their list. Unfortunately, in the US, I don't know of any testing labs that can give you an up or down on Aphanomyces eutyches. That's certainly something that we're working on trying to develop. There are labs available in Canada, however, that might be able to take a soil sample and help you determine if Aphanomyces is present. Now, the next environmental condition you need to be concerned about is last time you planted peas or lentils in the field. Was it a really wet year or was it a really dry year? If it was a really wet year, that really gave the pathogen a chance to multiply. In a wet year, you're getting production of swimming zoospores, and so you get exponential increase of the amount of the inoculum in your field. And I'll show you on the next slide what that will look like. If you've had an above average moisture year with the pulse in the ground, they're recommending you wait a little bit longer on that particular field before you plant a pea or a lentil. In this case, they're recommending 8 to 10 years. And then did you observe symptoms? So I'll show a little bit what that looks like, but essentially, did you observe some above ground, yellowing and patches in your field? And if you did, that's indicative that you have a root rot problem and you need to wait a little bit longer before you plant a pea or a lentil again. So this is available on the Saskatchewan Pulse Growers page. So if you just Google Sask Pulse Field Selection Guide, you should be able to find it. So this is an example of what can happen if you have one wet season. This is a field in North Dakota. These photos are courtesy of Julie Pashy, but this is actually one field. And so in 2011, we had a really wet season at the beginning of the year. Farmers got some of their crop planted, but then got rained out eventually during the planting season. So there was a lot of acres that went unplanted. This particular field, the grower was able to plant half of it to peas and then was rained out and wasn't able to plant the other half. These symptoms are from the next time peas were planted. So the healthy peas on the left are from where no peas were planted in that one year. And then the peas on the right are where the peas were successfully planted in that wet year. So it just really took that one wet year to cause this pathogen just to, increase exponentially in the field. And this was the result. So it can be very devastating following a wet year. And that's why those crop rotation recommendations are in place. So moving on to Fusarium root rot. This is what symptoms look like kind of at the early stages before root rot becomes a big problem in a field. This could be aphanomyces as well, just from the above ground symptoms. You can't really distinguish them, but essentially what you'll see is just these yellow patches that tend to follow where water moves through the field. So you can see it's kind of moving down the hillside, and then the symptoms, are increases in the bottom of the field where the water is sitting. However, this disease can become quite problematic, affecting a whole field. And depending on what Fusarium species are present, you can get Fusarium wilt as well. So all three pulse crops are susceptible. There are multiple Fusarium species involved. Likely there is some host preference, but this is definitely an active area of research. Fusarium solani, for example, we have the Forma specialis pisci. So that Forma specialis just means there's some host specificity, in this case to P. And that's the predominant Fusarium pathogen in the Palouse groin region. In surveys in North Dakota, we haven't really seen as much solani. We've been seeing Fusarium avinaceum and Fusarium oxysporum on P, and Fusarium oxysporum on lentil. So there might be some host specificity there. This work is ongoing. We believe that Fusarium avinaceum and Fusarium graminearum are so common on the root rot because we are rotating with wheat. Fusarium graminearum is the causal agent of Fusarium head blight in wheat. So we're in a no-till growing system. So all these Fusarium species are just going to be present. And, as you plant into the wheat stubble, there's the opportunity for the pathogen to reach the pea or lentil root as well. So Fusarium avinaceum has been frequently isolated from pea, both in North Dakota and in Saskatchewan and Canadian growing regions as well. And it's a major contributor to root rot on pea. And it seems like Fusarium oxysporum is the predominant root rot pathogen for Fusarium root rot in lentil. So obviously these Fusarium pathogens can be quite cosmopolitan, can affect multiple crops, but crop rotation is going to help limit all these other, pathogens that are affecting the crop. So a good crop rotation is just a way to reduce disease overall in the pulses. And Fusarium is definitely an opportunistic pathogen. And if aphanomyces is a problem, you're going to have more problems with Fusarium root rot as well. So you want to stick with your good 4-year crop rotation for peas and lentils. Seed treatments are available for Fusarium root rot. That's only going to give you early season protection. This pathogen can infect season long. But if you've got some Fusarium seed rot issues, that can help increase crop emergence. Similar to aphanomyces, early planting is recommended. Fusarium grows more aggressively under warm soil conditions. So if you're planting peas or lentils early, you're going to give that crop a chance to emerge and get going before it's infected. And similar to aphanomyces, there are no resistant varieties available. So I wanted to show some research comparing early versus late planting. This is a collaborative project with Michael Wunsch. He's a plant pathologist at the Carrington Research Extension Center, which is based in Carrington, North Dakota. So here I'm showing root rot severity and yield on the same axis. And this is a 2018 field trial where we inoculated with Fusarium. So the Fusarium infested grain went down with the pea seed. So in terms of root rot severity, the 2 later planting dates, so generally late April, mid-April is probably the earliest I'll plant pulses in our area, depending on soil temperatures. So April 26th is early, but not incredibly early. May 7th, May 16th is getting a little late. And you see root rot severity increases with the later planting date. In terms of yield response, there wasn't as dramatic a yield response, but the later planted peas did have decreased yield. Obviously, the yield response to the disease is going to depend on the severity of the disease. This is another trial. So this was done in 2020 in a grower field that had a history of Aphanomyces root rot, and Fusarium was present as well. So the early planting clearly holds true again for suppressing the root rot severity, a strong response. In this case, there was no yield response, but it was a drought year. And so probably the drought was the greatest yield limiting factor, not the root rot, but definitely decreasing the severity of the root rot with early planting by a good degree. So in all of these studies, we used watchdog temperature loggers, and we placed those at the seeding depth. So 2 inches seeding depth was what we used. And Michael analyzed this data and found that yield was optimized when soil temperatures were 45 to 50 degrees Fahrenheit in the week after seeding for peas, and 45 to 54 degrees Fahrenheit for lentils. Root rot was minimized when soil temperatures in the week following seeding were below 50 degrees. And establishment was problematic if you seeded below 45 degrees. So we had some issues with frost damage in the peas at the earliest planting date in that 2020 trial, but otherwise I didn't observe any problems. All right, so in terms of crop rotation, a 4-year or 6-year crop rotation might sound really long, but if you're new to pulses, this is really going to make sure that you can produce these crops for a long time without having a problem with the root rots. Where we've seen close rotations for a decade or more, some of these growers are no longer producing peas or lentils. So we finally have a good data set from North Dakota, again, thanks to Michael Wunsch and John Rickertson, to demonstrate what happens when you rotate, every other year wheat field pea. And so these are 2 different sites. Hettinger, North Dakota, that's southwest North Dakota, very dry area. And this particular field had no field pea production history prior to 2014. So the 3 treatments are a 2-year rotation, wheat, field pea, a 3-year rotation, 2 years of wheat, followed by field pea, and then a 6-year rotation, wheat, barley, canola, wheat, corn, field pea. And so you can see the plant population didn't change much. Typically, Aphanomyces isn't causing issues with the seed rot like the Pythium is. So no effect on establishment. If you look at root rot severity, you can see there's already a higher degree of root rot in that 2-year rotation after 4 years of the pea in a field that had no previous history of disease. Not a strong yield response yet, so just a 3 bushel difference between a 2-year and a 3-year rotation, a 1 bushel difference between 2 and 6-year rotation. But obviously with time, as the pathogen becomes more established, that yield effect is going to be stronger. And that, again, was that drought year where we had a lot of issues with the yield, not just because of disease, but also because of the drought. So then in the Carrington site, this was actually a field pea production, a seed production field. And they'd stopped growing field pea in it because the disease got too bad. So this was a perfect site for their study. Again, no effect on the plant population among the 3 different rotation treatments, but a strong effect on root rot severity. So 26% severity in the 2-year rotation and 8% severity in the 6-year rotation, with the 3-year rotation being intermediate. So even having 2 extra years compared to 1 year was beneficial. If you look at yield, the 2-year and 3-year rotation, not statistically different, but a strong increase in yield in the 6-year rotation compared to the 2-year rotation. So, the minimum we're recommending right now is a 4-year rotation, so a 3-year break in between peas or lentils. But if the root rot becomes established in your field, like in the Carrington site, a 6-year rotation could help remediate the issues you're having, with the caveat that this study was actually incorporating the early planting as well. So early planting, good rotation should put you in a decent position to deal with these root rots. So just a note on diagnosis, because so many things can cause yellowing above ground or even, bare patches in the field, it's important that you dig the plants or dig the seed to confirm that it is a root rot issue. I've gotten several calls where I go to the field and I dig the plants and the roots are completely healthy. And so that's a good place to start, is just looking at the roots. But note that the visual diagnosis is not sufficient to determine the causal pathogen. So you will have to send it into a diagnostic lab for them to perform isolations from the roots, look at the root under the microscope, maybe even perform some DNA testing to determine if a Phantomyces is present or, it's a fusarium issue. Something that we're hoping becomes available in the US, the research is ongoing. We're working on a grant proposal right now to fund this work, but we'd really love to have a comprehensive soil DNA test. This is a test result from Predict2B. It's a system currently available in Australia. Obviously, that's a little bit far to ship soil, but just to show you the potential of an assay like this. These are fields that I sampled in Northwest North Dakota, and you can see, 2 of these fields are positive for a Phantomyces eutyches. We can quantify the pythium level, the fusarium level, rhizoctonia levels. And these 2 fields that were positive for a Phantomyces, I called the growers and they said they did have really severe root rot issues. So having a predictive assay like this could really help allow growers to make field selection decisions, and hopefully is something that's going to be available here in the near future. All right, so I'm going to move on to foliar disease with a focus on ascochyta blight. I'm going to discuss this kind of in the context of chickpea because it's most problematic on chickpea. You can see ascochyta blight on peas and lentils, and it is a good idea to scout for it. If folks see it, they might make one fungicide application for the control of this disease in peas or lentils. But in chickpea, you can make 2, 3, even up to 5 applications of fungicide. So definitely a different management scenario in these different crops. It is host specific. So if you're rotating peas or lentils with chickpea, just note that your rotation length is going to be specific to the chickpea. It can affect all plant tissue. So you can see some of the symptoms on the pod, the stem, and the leaf. You get that characteristic bullseye lesion. And a lot of the loss comes from when you have these lesions on the stem and the whole plant will lodge over. So you can see severe disease in this plot photo I have here. This is a fungicide trial, and this is my untreated plot. So left unmanaged, it can be extremely aggressive. Obviously developing most quickly under high humidity, high rainfall conditions. So this past year we had a severe drought. Some growers were able to get away with not spraying their chickpeas just because, we really didn't have the moisture to get the disease going. So when grown under the appropriate conditions, this disease can be managed as well. So I just want to discuss the disease cycle a little bit. So this is the in-season spread, and this is one of the reasons why the pathogen can be so challenging to control. You see the lesions full of these pycnidium. They're spreading the spores in a cyclic manner throughout the growing season, and this spread is facilitated by rain and wind. So when we're trying to forecast the disease pressure, we kind of go based on the weather forecast. If we're having a rain event coming up, and you're at the end of your fungicide window, we recommend making a fungicide application. If it's dry, there's not been any rains recently, we know that this spread is going to be a lot less. But that said, you can have multiple cycles of disease spread within the season. It's very important that if you're controlling this disease, you make a fungicide application when you first see symptoms. Otherwise, you're going to get more lesions and more spread. So later in the secondary part of this cycle, so the cross-season spread, your pathogen is coming from infected seed or it's coming from infected residue. So whatever's left over in your field, we're in a no-till production system here. So we have the crop residue left over and then what's blown out the back of the combine. You need to give the pathogen time in between growing seasons to become no longer viable. And so, we recommend a minimum 4-year break from chickpea to allow that pathogen to fully, decompose. And then in terms of the seed, you can submit your samples for seed testing to determine the level of ascochyta blight pathogen on your seed. So there is no silver bullet for ascochyta blight. It really takes a stacked approach. So an integrated pest management approach, start with that seed testing, make sure you're starting with healthy seed. I'll show some data about variety selection. So some varieties have pretty good tolerance. Your crop rotation to allow that pathogen to break down. There's some data showing that the increased row spacing, so 20 to 40 centimeters, the 40 centimeter row spacing did have reduced disease severity. Which is perhaps why intercropping has been a practice that's been adopted by farmers to manage ascochyta blight and reduce or eliminate the need for fungicides for some farmers, depending on what their goals are. And then if you are using a fungicide, I know this is organic group, so probably many of you will not, just scouting and making sure you're staying on top of the pathogen. So locally, we have some seed testing resources available. I did mention that the Pulse Crops Diagnostic Lab is regional. It can take samples from around the US. So the seed testing is anywhere from $160 to $180. You need 1 to 2 pounds to submit the samples to the lab. And different states have different tolerances for ascochyta blight on the seed. In North Dakota, a level of 0.3% or higher is considered very high for chickpea. And in Montana, there's no tolerance for ascochyta blight on chickpea if it's sold as seed. It should be fine in the commodity market, but this is if you're using your seed, trying to sell your seed as a seed source. And generally speaking, the pea or lentil tolerance levels are higher than they are for chickpea. So this is some variety testing data, 2 years of data from the Williston Research Extension Center. Both years were drought affected, so we had anywhere from 4 to 5 inches of rainfall season long. Drought was definitely more severe in 2021, but even so, we're seeing differences in disease severity and yield among the different varieties tested. So CDC stands for Crop Development Center. These are Canadian varieties. So Frontier, Leader, and Orion, definitely among our highest yielding varieties, and also with some of the lowest disease severity values. So this is ascochyta blight rated mid-July based on the percent of the crop canopy exhibiting disease symptoms. And so overall, you can see disease is really low. Even so, the 2 varieties that were most susceptible, Royal and Sierra, had pretty high disease severity compared to the other varieties in 2020, slightly elevated disease in 2021, but a very strong difference in yield. I think there's also an effect of some drought tolerance. I believe CDC Frontiers is slightly more drought tolerant, but you do see a benefit to growing a disease-resistant variety, even in a season with very low disease pressure. ND Crown, I wanted to mention, that's going to be a new release available from NDSU. You can see it has a comparable disease package to the Canadian varieties with comparable yield as well. So intercropping, Dr. Claire Keene was doing some work with intercropping chickpea and flax at our research farm. And this data is from Dr. Mike Osley. He's a research agronomist at the Carrington Research Extension Center. So if you're interested in using intercropping as an approach for minimizing ascochyta blight in chickpea, obviously you have to have a market for flax, first and foremost. Some people are also using a mustard as an intercrop partner for pea. But if you're using conventional weed management approaches, flax is definitely a more convenient partner for chickpea. So the increased flax yield is going to depend on the growing season. These are similar rates, seeding rates of chickpea in regards to the ratio of chickpea to flax, but different growing conditions in 2 different years resulted in strongly different flax yields, which are going to impact your chickpea yield. So just note that regardless of the disease control, because you're growing 2 crops together, you're going to have slightly lower chickpea yield. So there's 2 fungicide treatments here, and comparing chickpea alone to chickpea intercropped. So in 2019, there was a strong reduction in yield compared in the chickpea alone compared to the intercrop partner, over 1,000 bushels compared to 255, pounds per acre. But if you look at the field without any fungicide, the difference is a lot less dramatic. So if you're not able to use fungicides for control of chickpea, intercropping can definitely be an approach you'd want to try. And so if you look at the ascochyta severity in 2019, there's a 50% reduction in ascochyta severity. And so that's not just going to affect the current season's crop, but also, your rotational interview, how much pathogen is left for the next time you see chickpea. 2020 results are definitely more promising. This is a drier year, lowered the flax yield, which probably caused less of a negative effect on the chickpea yield. So if you're comparing the chickpea alone to the chickpea intercropped, still a reduction in yield, but not quite as dramatic. You still have, some chickpea there. Interestingly, the intercrop chickpea had similar yield between whether you had a fungicide application or not. So the disease control from the intercropping in this case was comparable to a 2 times fungicide application. In terms of ascochyta severity, similar levels in ascochyta severity where the fungicide was used, but a strong reduction in disease severity where fungicide was not applied. There's a lot of ongoing research with these different intercrop partners, but if you're in an organic system, intercropping chickpea with flax could really be useful for you. I'll just touch on the fungicides really quickly, just in case there's anyone on the call who's interested in that. Just to note that FRAC 11 strobilurine fungicides are not effective for chickpea ascochyta blight in North Dakota. The pathogen is resistant. It's resistant in other growing regions as well. So if you're repeatedly applying these fungicides, chances are the pathogen will become resistant in your growing region as well. The P pathogen, one of the P pathogens is also, we've noted some fungicide resistance to these strobilurins as well. So FRAC 3, FRAC 7 are the FRAC groups we're recommending, so the DMI fungicides, SEHI fungicides. You need to rotate these FRAC groups so that we keep these fungicides available. And then tank mixing with a multi-site mode of action, broad spectrum chlorothalonil, has shown a really excellent control. You're also lowering the risk for development of resistance. So we tank mix that with Proline, and we've shown excellent control from that. And that's, again, an ongoing project with Michael Wunsch. Your application is going to vary based on whether you're seeing signs of disease. So if you're not seeing any disease, we usually apply early flowering to apply the product before the canopy closes. And then repeating every 10 to 14 days based on disease and also the weather. So the most I've done here is 2 under non-irrigated conditions, just because we're so dry. There are some resources available. So NDSU has a growing chickpea in North Dakota guide. And then we also have the North Dakota Field Crop Plant Disease Management Guide that's released every year. And that lists all the fungicides that are labeled both as a seed treatment and foliar fungicide in all 3 of the pulse crops, as well as the other crops we grow here in North Dakota as well. So just to summarize our approach to disease management in pulses, you're really starting from your crop rotation. So they need to be long, minimum 3 years in between peas or lentils, and primarily in that case for the root rots, but that will also manage your foliar disease as well. Minimum 4 years between chickpea, most importantly for that ascochyta blight management. One organic grower I know in Montana has had really good luck incorporating alfalfa into his crop rotation to really extend it out to about 7 years with 3 years of alfalfa. So it's a wheat, 3 years of alfalfa, wheat, pulse, wheat rotation, where you're getting your nitrogen from the alfalfa and from the pulse. And so that really cuts back on your fertilizer needs for the wheat. And a lot of that can come from the pulse crop and the alfalfa then. Just making sure that you're incorporating an Aphanomyces resistant alfalfa. You could also use a brassica green manure cover crop in there in your rotation, both for the disease management and also, taking a year out. Obviously, all of these might be more economically feasible if you have cattle or work with someone with some livestock. So you can, turn these cover crops or forages into, an income stream as well. And there's some more resources available from NDSU and from the Pulse Crops Working Group. So this is the Pulse Crop Production Field Guide. That will have peas, lentils, and chickpeas in it. There are diagnostic series available for pea and lentil online. And then this dry pea and lentil root rot management guide. That was published in 2020. And then if you aren't already a listener, the Growing Pulse Crops podcast will discuss all of the different things I discussed today. There's several episodes on intercropping if you're interested in that practice. So I highly recommend you check it out, growingpulsecrops.com or anywhere you download your podcasts. And with that, I'd like to acknowledge the contributions of Dr. Osley and Wunsch to this presentation. Haney and Dimitri have done a lot of the work that I presented today. And just to note that we are recruiting. So if you have any undergrads interested in a summer internship, we're also hiring a research technician right now. And I'd be happy to take any questions. Thank you.
Great. Thank you, Dr. Kalil. We really appreciate it. That was wonderful.